T cells receptor was co-discovered by Dr. Takwa Mak in 1984. Born in southern China in 1946 to parents who were silk merchants and raised in Hong Kong, Mak and his family moved into the United States during mid-60s and was persuaded by his mother to attend University of Wisconsin. His interest in life and chemistry led him to studying biochemistry and biophysics at the university. After finishing his degree at Wisconsin, Mack moved to Canada to become his doctoral studies at University of Alberta, Edmonton. In in the early 70s, he earned his PhD in biochemistry from the University of Alberta. After he obtained his degree, Mack moved to Toronto and became a Canadian citizen. In 1984, Mack discovered a T cell receptor with Mark M. Davis, identifying the receptor in mouse. This work on cloning T cell receptor genes as of 2005 has been cited nearly 1,200 times. His other breakthroughs include making first lab-made genetically modified mice which utilized in science labs around the world. In 2004, Mack became the director of Advanced Medical Discovery Institute and the Campbell Family Institute for Breast Cancer Research. In 1984, he has been a professor in the Department of Medical Biophysics and Immunology at the University of Toronto.
So you might be asking, what exactly is a T cell and what does it do? Well, the T cell is a type of lymphocyte, which is a subtype of white blood cells. It plays a central role in the cell-mediated immune system. The T cell can be distinguished from other lymphocytes, such as B cells, by the presence of a T cell receptor on the cell surface. They are called T cells because they mature in the thymus. The thymus is a specialized primary lymphoid organ of the immune system. There are four main types of T cells: helper, killer, memory, and suppressor T cells. Helper T cells have an important role in immune tolerance. Their main job is to activate B cells to release antibodies to fight the infection, and they activate killer T cells to perform their function. Killer T cells, or cytotoxic T lymphocytes, are part of cell immunity and perform cell-killing effects. They usually target self-altered cells like virus, bacterially infected, or cancerous cells. Suppressor T cells work like a brake in a car. Once the infection has been dealt with, suppressor T cells stop the function of killer T cells and also suppress autoreactive T cells. Memory T cells persist long after an infection was resolved. After infection, they rapidly proliferate to large numbers of effector T cells upon re-exposure to their antigens, providing them with memory so that if the same antigen were to strike again, they could deal with it in the exact same way. Some other T cells include effector T cells, natural killer T cells, and gamma delta T cells. So why is this research so important? Before his breakthrough, researchers knew T cells attacked viruses and bacteria, but no one knew how they worked. Mack was able to describe T cells as hitmen, telling friend from foe and ordering attacks on pathogens. They were able to determine between good and bad using receptors which were discovered by Mack. The surface of the T cell are covered with receptors that identify cells containing pathogens like invading bacteria. Mack's discovery on T cells receptors was important because T cell receptors are essential to your health. If the receptors don't recognize pathogens, then the T cells can't do their job to fight it off, and you most likely die. Understanding how T cells work has allowed us to develop new drugs for fighting infection, autoimmune disorders, cancer, and post-transplant rejection. In a recent study, scientists have claimed extraordinary success with engineering immune cells to target a specific type of blood cancer in their first clinical trials. In one study, 94% of participants with acute lymphoblastic leukemia who were only given 3 months to live saw symptoms vanish completely. Patients with other blood cancers had response rates greater than 80% and more than half experienced complete remission. So how does this process actually work? Well, to start, a sample of the patient's T cells is taken from their blood. In the lab, these cells have genetically modified antigen receptors added to them so they can seek out certain cancers. Modified cells are allowed to multiply before being infused back into the patient, where the new antigen receptors allow them to target and destroy the cancer cells.